So we're going to talk about clinical trials for medical device approval. And the subtitle is "The Debate Rages On." 1 lakh rupees for anyone who could tell me what movie this came from. I heard somebody say it. So device regulation in the United States. In the United States, the first formal legislation to regulate devices came about in 1976, many, many years ago. 3 classes were created based on device risks: Class 1, Class 2, Class 3. And Class 3 requires a lot of research. You have to prove to the FDA that you're safe and efficacious. Class 2, the moderate to high, could fall into this 510K pathway, and we'll talk about that. 510K is designed as an alternative to the very stringent PMA process, where you have to start your research from scratch, because it applies to technologies that are modifications of commercially available devices. So if you have a device that's very similar to something that's already been approved, you don't have to go through all the research to show that it's safe and efficacious to get clearance. And so most devices that fall under this sort of expedited pathway fall under this Class 2, moderate to high risk. And this is all based on what we call substantial equivalence. Now, worldwide, the rest of the world has adopted a similar policy. You could be in the FDA, the US, where the FDA has a 510K. India has a predicate pathway. China has what's called the same type. Korea, Japan, they all have very similar predicate device sort of comparisons to get to market. In India, where we are now, approval through the Central Drug Standard Control Organization, CDSCO, where manufacturers just need to demonstrate that their device is substantially equivalent to a predicate device. In India, you could also bypass that completely if the device has already been approved by one of these reference countries and the device has been successfully marketed for at least 2 years. So this predicate device comparison is a very nice way to get your technology, your new technology to market. Substantial equivalence is defined in terms of similar intended use and technological characteristics, or if any of these two are different without any anticipated effect on safety or efficacy. In the US, over 175,000 medical devices and 18,000 manufacturers have used the 510K process. The 510K really only requires post-market surveillance to ensure safety. This is either done through self-reporting by companies, and in 1990, the FDA allowed clinicians and hospitals to report as well. Now, there are potential problems associated with this because exception has become the rule. 90% of medical devices in the US are cleared via the 510K process. Less than 1% of all applications submit any clinical data, and any data requested by the FDA occurs in less than 10% of all approvals. And there's no clear regulation on predicate devices. 20% of the predicate devices that led to clearance were out more than 10 years, and the predicate themselves at the time of the 510K process were sometimes recalled afterwards. So this could lead to people getting very concerned and upset. This is a perfect example. This is a letter written to the FDA by a series of physicians in the US, authored by Lonnie Armas at Johns Hopkins. And this is in regard to microwave ablation. And they wrote, "We have substantial concerns which temper our enthusiasm for this technology and believe that the current available data supporting bronchoscopic ablative therapies in the lung are immature, concerning as illustrated by the recent deaths that Kyle mentioned earlier in a prior trial investigating use. We are unanimous in our opinion that before any further investigative attempts in this space are pursued, the FDA should consider a temporary suspension of the current 510K clearance for bronchoscopic ablation and go on to a PMA." And this was signed by a lot of people you may recognize. So what's the answer here, right? Is the answer that we do away with the 510K, or do we do more research to bring out a device? And it's created divisions, certainly within our organizations and our groups. So here's a real story that I think is illustrative of some of the issues. And this is neither for or against. This is just a real story that I want to use to illustrate. And Intuitive Surgical is the one that we're going to use. This is the original 510K application for the Da Vinci surgical robot, right? So, you go out and have robotic surgery. This was from 1997. You could find this on the internet. It's public record. June 20th, 1997. Everybody knows what a surgical robot is. What were the predicate devices? One was computer motion laparoscopic position. So basically it was a laparoscopic device that had a positioning accessory, which allowed you to move the laparoscopic scopes around. And at the tip of it was a stapler. This is the predicate device for the Da Vinci robotic system. Look what happened. The Da Vinci got approved, it got brought to market, and you could see what happened in the next 20 years after it was brought to market. This is just data from 2010. You could see the install base in the United States goes up to almost 3,000 robots. Even in the non-US, the rest of the world, 1,500 robots out in the rest of the world, and you could see the procedure volume going up significantly. Intuitive in 2023, it placed 1,370 surgical systems, an increase of 8% from the year before. So this keeps going up. The fourth quarter 2023 worldwide Da Vinci procedures grew 21% over the prior year. It's pretty significant when you think about it. If you look at where they got their cases from, well, you could see very clearly that they took cases away from open and laparoscopic surgery. You could see on the chart on the right, opens kind of went away as laparoscopics grew in popularity, and as soon as robotics were introduced to a hospital system, laparoscopic surgeries went down, robotic systems started to go up in their place. Okay, well that's great, right? But this happened despite no evidence of efficacy or equivalence or superiority. And if you look at cost, doing a robotic surgery was more costly than doing a laparoscopic surgery. So think about this. You have a device that costs over $1 million US dollars. You bring it into a hospital. You start doing procedures, taking away from laparoscopy, which costs more, and no proven benefit above and beyond. So if an editorial in the New England Journal of Medicine, if robot-assisted procedures replaced all conventional procedures, an additional $2.5 billion would be spent in annual healthcare costs in the USA. It's a lot. Now, how does that really affect us? Well, let's talk about benign resection just for a second. At Washington University in St. Louis, they demonstrated an increased rate of benign resection rates, okay, once VATS was instituted. The benign rate was double in VATS as it was versus open. Turkish data showed that robotic segmentectomies demonstrated a benign resection rate of almost 30%. A recent study out of Mass General, Harvard, in the US showed a 15% benign resection rate. So this implies that maybe robotic surgeries, because they're easier to do, at least for the surgeon, more sexy to do, that they promote perhaps non-therapeutic thoracotomies because risk goes down, let's take the risk and go in and do it. So again, how do we apply that to robotic bronchoscopy, right? Which is the big sexy thing in the US right now and probably will be the big sexy thing in Europe and Asia over the next several years. That hype is sort of taken over, market forces are dictating care. People are spending a lot of money on high-end technology that may not be better than what we already have had. And at least in the US, $500,000 for a base robotic system, $300,000 to $1 million, depending on what kind of imaging you want to use, $1,500 to $2,000 per case cost. I mean, these are staggering numbers when you think about it. And all this does is rely on anecdotal data to support procedural volume. There's some data that's emerging, and you're going to see it later, and you saw some from Kyle, you're going to see some from Mike Pritchett a little bit later. But again, this kind of data relies on arbitrary, unproven endpoints to assess outcomes and success, which perhaps may lead to more procedures on smaller and smaller nodules with no discernible endpoint, perhaps leading to unnecessary procedures that will just overtake standard surveillance, increasing risk, increasing cost. So again, a lot of this is not based on data, it's based on my anecdotal experience. And why is this bad? This is what market forces are, right? Market forces are not so much developed on data, but they're developed on your anecdotal experience. So what is the answer? Should we get rid of the 510K process altogether? This is what some people would suggest. More clinical trials, this is again what some people would suggest. Perhaps creation of pragmatic comparative efficacy trials, or creation of cost and resource utilization trials. Maybe a total revision of outcomes definitions and terminology. Maybe a post-approval quality outcome tracking and reporting. All these things are good ideas, but you could poke holes in all of them. Perhaps, maybe we should just let market forces dictate. Because after all, it's free market, right? We want to do what's best for our patients. We should be able to spend our money however we choose. I don't know what the right answer is. So let's talk about these over the next just few minutes. How much and how long does it take to do these kind of trials? Clinical trials are enormously expensive. A 300-patient efficacy outcome trial across 10 sites in the US could cost over $20 million. It's expensive. And it takes time from start to finish to a publication. It could be somewhere between a 3 and 5 year cycle. So imagine you come out with a new technology, you've already spent millions of dollars developing it. Now you have to spend another $20 to $30 million to do the trial, and now you have to wait 5 years before the FDA will even consider it. It's too much. What about pragmatic cost efficacy trials? Well, they're certainly easier to do, may cost less, but for cost to be less, the device would need to be commercially available. So we kind of are repeating on ourselves, right? The data certainly wouldn't be as robust. Clinical endpoints, very difficult to define. And by the time it's done, systems are already purchased, they're already out there, they're already being used. So what's the point of doing the study in the first place? Who would do them, right? Where would we do them? How would we do these trials? Well, here's just a timeline of the superdimension, right? The Lumicite. And you could see very, very quickly, over sometimes a span of months to years, the technology advanced. So if I did a trial in 2012, right, which is the Navigate trial, by the time 2015 came around, that Navigate trial was almost irrelevant because new technology had replaced what they were already studying. So technology moves faster than clinical trials could go. Oh, what about who could do it? Who's doing these trials? This is one of the more famous studies that are out there. This is the Acquire Registry. Apparently the best of the best in the United States and the world doing navigational bronchoscopy. And look what they showed, that if you didn't use EMN or radial EBUS, your diagnostic yield was 63%, EMN alone, 38% yield, navigational bronchoscopy with rebus, 47%. So it kind of proved the exact opposite of what navigational bronchoscopy should do. So were the right people doing this? Did they know what they were doing with the technology? The other thing that we have to contend with is diagnostic yield, right? Diagnostic test performance is really a combination of many different things, some of which are controlled, some of which are uncontrolled. It's really a combination of the performance of the technology, the operator performance, and then many other things that you really can't control. All these things lead into diagnostic yield. Diagnostic yield is defined in many different ways, and it's been problematic in the literature. There's very strict diagnostic yield definitions, which we, I think now, thankfully, use. But there's studies out there that use very liberal definitions of diagnostic yield as well. We'll talk about this more tomorrow in a talk that I have on diagnostic yield. But again, these could be very, very different. If you look at, this is just an experimental cohort, but again, depending on how you define diagnostic yield, you could have yields that are 20% different. You could go from 89% down to 66%. Same patients, same outcomes, defined differently, huge differences in diagnostic yield depending on how you define it. The Navigate trial, which is one of our landmark trials, they reported out a 72% diagnostic yield, but if you use a strict definition filter, their yield drops to about 54%, which is a coin flip. A coin flip which is free, navigational bronchoscopy which is not. Look at all the robot trials out there in the literature. Their diagnostic yield has been definitions has been all over the place, from non-existent to intermediate to strict. So how do you compare studies? One study that says I have a 98% diagnostic yield but used a very liberal definition of diagnostic yield. Well, cancer prevalence also, right, also impacts this. In the United States, it's a huge problem because in the shaded areas in the United States, there's a significant amount of benign fungal disease causing nodules. So if I'm using a robotic platform to diagnose a lung nodule, say in New York, everybody knows where New York is, that side, versus where, for instance, me and Kyle live, which is in the gray area, right, where benign disease is much higher, it's hard to compare those two populations. And you could see the differences that you'll get using different types of yield definitions on the graph behind me. What about nodule size? Nodule size impacts cancer risk too. So how can I take two studies, one of which used a patient cohort of 20 millimeters or larger, another of which uses a smaller nodule cohort, right? We expect those yields to also be different. So how do we do these studies? How do we compare technologies when things are all over the place the way they are? And what are the correct endpoints? Is diagnostic yield really the right measure of success, right? Should we consider other endpoints, such as diagnostic accuracy, such as tool and lesion? I would argue that a radiographic tool and lesion, if I could put a needle out into a nodule, prove that the needle is in that nodule radiographically, I think that I've done exactly what the technology prepared me to do. Everything else after that is completely separate from the technology. How I do the biopsy, how the slide is prepped, how the pathologist reads it. So I would perhaps argue that tool and lesion may be a better surrogate than diagnostic yield. Again, these aren't things that are discussed right now in our medical literature. So, you know, in the US we have this term, don't compare apples with oranges. And the same thing goes for the clinical trials. So if you can't compare these clinical trials, what do you do? How do you do this? And how do you mitigate this when you want to bring a device to the FDA for approval? So, in my summary opinion, in Kyle's font, in my opinion, I think clinical trials prior to FDA approval are expensive, time-consuming, and they're very difficult design in a variety of ways. And as a result, vulnerable, at-risk, and really in-need patients who could benefit from technology are prevented from obtaining potentially beneficial diagnostic therapeutic procedures. The 510K process, certainly not perfect, but to expedite a new technology that could help people to the market, I think is the best way of getting cutting-edge technology to the market. We should just get things to the market and have robust post-marketing surveillance, especially for safety, which is critical. You could do these clinical trials, word of mouth, however you want to do it, but all these things influence adoption. And trust me, if we bring a technology to the market that doesn't work, nobody's going to use it. If you bring a technology to the market that works extraordinarily well, people are going to talk and they're going to say, hey, I'm doing really great with this. You should get this. You should get this. And that's, I think, how robotic bronchoscopy has just mushroomed in the United States. And I think that's how things should be. Again, just my opinion. But again, I'll end this on the famous economist John Keynes, who said, markets are moved by animal spirits. That's us. It's us people in this room, and not by reason. Thank you.
Great talk, Joe. I mean, I think you've highlighted beautifully the dilemmas between research, time over research, but at the same time, I think the counterpoint would be that if we don't do research, for example, a very simple scenario for ion bronchoscope, robotic bronchoscope, is that we could probably sample more than 50% of the nodules that they're recommending with very cheap, effective transthoracic guided CT guided biopsy. So really, to get this incremental progression in medicine, it takes time, but that way we get the right answers.
Yeah, look, I agree with you, and that's why I showed the data from the Da Vinci robot, right? It's a surgical robot, has never been proven to be superior to laparoscopic procedures, but way more expensive, and you know, I think there are analogies to robotic bronchoscopy that could be applied and lessons learned. That being said, I don't know how you right now go about fixing that problem.
So it's not. So I think the answer, in my view, is that it's not that we should cut research. We need to reduce the time it takes
Yeah.
And the only way to do that is to choose the correct endpoints.
Correct. That's it.
Yes, there is a question and then,
Is that Dr. Toma? I can't see.
yeah. I have just a question waiting for Tom.
Okay.
Thank you, Joe, for
You're
your
welcome.
presentation. My question is regarding the endpoints.
Yeah.
So we are dealing with very small nodules,
Yes.
and the biology of these nodules, even if they are tumors, are not yet well known. So what can we define the endpoints when we are dealing with these small nodules? Is it better to wait and see, to have some other elements to detect biologic behavior, or just to biopsy them?
So if you come into some of our chat groups, you'll see this argument. There's one going on just yesterday. Not an argument, but a discussion. Look, these robots, the robotic bronchoscopy in the U.S., when paired with imaging, really, Pavlov in the UK is doing this. You could really get down and get a tool in a 7 to 8 millimeter nodule very easily. And you get tissue. You could get great tissue with needle, forceps, cryoprobe. But just because you can doesn't always mean you should. I think that you have to be a responsible physician, do the right procedure on the right patient. Maybe look for radiographic progression before you do things. Doing something and having the wisdom to do something are two different things. So it's a great point. And how you regulate that, I don't think you can. I think it just has to be on an individual basis. There's in the U.S. we call them quacks, right? They're quack doctors, everywhere. I can't control that a doctor 10 miles down the road is not as good as me. It's just the way it is. You have to, I think that the only way to kind of move forward is you do your procedures, you track and report your quality, your outcomes, and let the patients decide where they want to go.
So I hope the microphone works. I'm Tudor Toma. I work in London. I've been involved many years ago with the endoscopy.
We cannot understand, can we?
Just right here.
Echo.
No, just do it here.
I've been involved with the endoscopic lung volume reduction from the very beginning,
Yeah.
and it took 20 years for this procedure to be approved. The main reason at the beginning was that the FDA was requiring an endpoint for the trial, which was not the right endpoint. This procedure was called volume reduction, and they were looking for FEV1. So what does the American community do for synchronizing the FDA with the realities of clinical medicine? That's the first question. And I have a comment regarding your fonts on the PowerPoint.
Oh, thank you.
So the way to solve that problem, you go into preferences, you save the file with embedded fonts, and it will come up
Okay,
with,
thank you.
with a good
Did you get that, Hogarth? Having worked with the FDA myself, they're very, very difficult. We could talk for hours on the FDA. They don't have the right experts on their experts panel. When you go to FDA and you negotiate with the FDA for a robotic bronchoscope, they may have an anesthesiologist, a pediatrician, and nothing against them, and an ENT surgeon on the panel. They don't understand what we do, so it's very, very difficult. Look, I showed you in my talk, I don't think diagnostic yield is the best outcome because our technology to get to a nodule does that. It gets you to the nodule. After that, the cytology interpretation, how you process your sample, that has nothing to do with the technology. I would argue that the robot exists to take me to the nodule. I mean, there's surgeons here. You go and do your wedge resection, right? And if you do a wedge resection and it comes back, non-diagnostic or benign, is that your fault? No. I mean, if the pathologist isn't good reading it, is that your fault? No. We don't judge you by the pathologist. We shouldn't judge the technology by that. So that endpoints I think are incredibly important in how we move forward. I just think we're using the wrong endpoints. It's just my opinion, but it's hard to tell a patient that, right? Yeah, so.
So thank you. I think in sake of time we have to stop
Okay,
here.
thank you.